Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today, we're going to be covering Chapter 2 in our MCAT Biochemistry series, and this chapter is titled Enzymes. Here we're going to cover the following objectives. First, we're going to talk about enzymes as biological catalysts. That means we're going to look at what enzymes do, how they affect reaction rates without being consumed, and the different classes of enzymes based on what types of reactions they catalyze. Second, we're going to look at the mechanisms of enzyme activity. So that includes how enzymes interact with substrates, the different models that explain enzyme substrate binding, and the role of helpers like cofactors and coenzymes that assist enzymes in doing their job. Third, we're going to dive into enzyme kinetics. So this section will focus on how enzyme activity changes as you vary the substrate concentration. We're going to go through the Michaelis-Menten equation, Lineweaver-Burke plots, and the concept of cooperativity, which explains how some enzymes behave differently when more than one substrate is involved. Fourth, we're going to explore how enzymes are affected by their environment. Factors like temperature, pH, salinity, these can influence enzyme activity. Finally, we're going to finish with the regulation of enzyme activity. We're going to go over feedback mechanisms and different types of enzyme inhibition, including competitive, non-competitive, and uncompetitive, as well as irreversible inhibition. We're also going to discuss how some enzymes are made in an inactive form and then they're later activated when needed. With this introduction in place, let's begin with the first objective and talk about enzymes as biological catalysts. Enzymes are proteins that catalyze reactions in the body. A catalyst is something that increases the rate of a chemical reaction, but without being consumed in the process. That means enzymes participate in reactions, they help them proceed more efficiently, and then they're ready to go again. So they're unchanged at the end of the reaction, and they can be reused over and over and over again. One of the most important things to remember is that enzymes affect kinetics, not thermodynamics. In other words, enzymes make reactions go faster, but they don't change whether a reaction is favorable or not. So they don't influence the overall energy change of the reaction, and they don't alter the position of equilibrium. They just help us get to equilibrium faster. Let's now walk through the essential feature of enzymes. These are foundational for biochemistry, and they're highly testable concepts on the MCAT. First, enzymes lower the activation energy of a reaction. The activation energy is going to be the energy required to reach the transition state, which is the high energy unstable state that occurs right before the reaction converts reactants into products. And by lowering this barrier, enzymes make it easier for molecules to undergo transformation. Because of this, enzymes increase the rate of the reaction. The reaction proceeds more quickly because more molecules can reach the transition state in a given amount of time. But keep in mind, speeding up a reaction doesn't change how much product is formed at equilibrium. It only changes how fast that equilibrium is reached. And that leads us to our next point. Enzymes do not alter the equilibrium constant. The equilibrium constant reflects the ratio of product to reactants once the reaction has stabilized. Enzymes do not shift that ratio. They just help the system reach it more efficiently. Another key property is that enzymes are not changed or consumed in the reaction. They interact with the substrate, they facilitate the conversion to product, and then they return to their original state. That makes them reusable catalyst, which is really critical for sustaining metabolism. Enzymes are also sensitive to their environment, particularly to pH and temperature. Each enzyme has an optimal range where it functions best. And if conditions deviate too far from that range, so if the pH becomes too acidic or basic, or the temperature becomes too high or too low, the enzyme can actually lose its structure. And since structure determines function, this usually means a loss of activity. It's also important to understand that enzymes do not change the overall free energy of a reaction, which we represent as delta G. So a reaction that is non-spontaneous, meaning it has a positive delta G, it will still be non-spontaneous even in the presence of an enzyme. Enzymes, they just help favorable reactions happen more quickly. But again, they do not change the overall free energy of a reaction. Finally, enzymes exhibit specificity. So each enzyme is tailored to recognize and act on a particular molecule or group of closely related molecules known as substrates. And this is called enzyme specificity. The enzyme binds to its substrate, it catalyzes that reaction, and then it releases the product. If the wrong molecule is present, the enzyme simply does not act on it. So to sum up, enzymes lower activation energy, they speed up reactions, they aren't consumed, they do not affect equilibrium or free energy, they're sensitive to conditions like pH and temperature, and they're highly specific for their substrates. Now, these are points I wanted to start off with early in the lesson, and they will be reiterated throughout the rest of the lecture. And that is because they are super important to understand. But now that we've covered the core principles of enzyme behavior, let's move into talking about how enzymes are classified. This is all about connecting the function of the enzyme to the type of reaction it catalyzes. Enzymes, they are grouped into 6 major classes. Let's go ahead and walk through each one. First, we have oxidoreductase. These enzymes catalyze oxidation reduction reactions, which are reactions that involve the transfer of electrons. These are common in energy metabolism, especially when molecules are being broken down or built up. For example, enzymes like dehydrogenase and reductase, they fall into this category. And a classic case would be transferring electrons from NADH to another molecule during cellular respiration. Second, we have transferase. These enzymes move a functional group from one molecule to another. A common type is kinase, which transfers phosphate groups. And another example is aminotransferase, which plays a role in amino acid metabolism. Third, we have hydrolase. These enzymes catalyze hydrolysis reactions, meaning they break a bond using water. They split one molecule into 2 by adding a water molecule across the bond. Fourth, we have lyase. These catalyze the cleavage of a single molecule into 2 products. Now, these cleave bonds without the use of water or redox chemistry, and they often generate a double bond or a ring structure in the process. Again, I want to emphasize, the key here is that they break bonds without water. Fifth, we have isomerase. These enzymes rearrange the bonds within a molecule to form an isomer. So in other words, the molecular formula stays the same, but the structure changes. A common example is phosphohexoisomerase, which plays a role in glycolysis by rearranging glucose into fructose derivatives. Finally, we have ligase. These join together 2 large similar molecules, typically using ATP. Now, you're going to see these in DNA replication and repair, where they connect segments of nucleotides. And these reactions generally require an input of energy, which is why ATP is usually involved. To remember these 6 classes, use the mnemonic Lil HOT. L stands for ligase, I for isomerase, L for lyase. Then the H stands for hydrolase, the O for oxidoreductase, and the T for transferase. Each class tells you something about the enzyme and what it's doing at the molecular level. And understanding these classification systems will help you recognize enzyme names and predict what kind of reactions they're involved in. Now, before we move on, I want to circle back to one of the most important effects enzymes have, their impact on activation energy. To understand this, we need to first distinguish between 2 types of reactions based on free energy, or delta G. An endergonic reaction is one that requires energy input to proceed, so its delta G is positive. On the other hand, an exergonic reaction is one that releases energy, meaning its delta G is negative. Now, here's the key point. Regardless of whether a reaction is endergonic or exergonic, there's still an activation barrier that has to be overcome. And that is the energy required to reach the transition state, which is the unstable high energy intermediate between the reactants and the products. If you look at this graph, the black curve represents the activation energy of the uncatalyzed reaction. But if there's an enzyme present, we see the dotted orange curve. It reaches the same final product. The difference is that the peak is lower. That's the enzyme lowering the activation energy and making it easier for the substrate to reach the transition state. Notice that the overall free energy change remains the same. It's still just the difference between the starting point and the end point. What enzymes do not affect the delta G. What they do is make the reaction happen faster by stabilizing the transition state or making it easier to get there. Then the follow-up question to that is, well, how exactly do enzymes do this? What's happening at the molecular level to lower the activation energy? And that brings us to the next objective, the mechanism of enzyme activity. Enzymes can speed up reactions in a few key ways. They might provide a favorable microenvironment. So, for example, they can create a pocket with the right charge or pH to facilitate the reaction. They might stabilize the transition state and lower the energy required to reach it. Or they might simply bring the reactive groups of the substrate closer together and position them just right for the reaction to occur in the active site. Each of these strategies relies on one critical interaction, the binding of the substrate to the enzyme. The molecule that the enzyme acts on is called the substrate, and the point at which it binds is called the active site. This interaction forms what's known as the enzyme-substrate complex. This is a temporary association that's essential for catalysis to occur. You can think of the active site as a specialized pocket on the enzyme that's precisely shaped to recognize and hold the substrate. It's at this site that the enzyme performs its catalytic function, whether that means breaking bonds or forming new ones or rearranging atoms. Now, there are 2 main models we use to describe how the enzyme and substrate interact. The first model is the lock and key theory. This model suggests that the enzyme's active site is already in the exact shape needed to fit the substrate, just like a key fits into a specific lock. The implication here is that only the right substrate will fit perfectly, and binding happens without much adjustment. And while this model helps convey the idea of specificity, it doesn't account for the flexibility we now know enzymes often have. And that leads us into the second model we use to describe how the enzyme and substrate interact, that model being the induced fit model. This model tells us that the enzyme undergoes a conformational change when the substrate binds. So instead of the active site being a perfect fit from the start, the enzyme actually molds itself around the substrate to create the ideal environment for the reaction. This model better explains how enzymes can be flexible and dynamic in their function. Now, both models agree that enzyme substrate binding is highly specific, but the induced fit model better reflects the fact that binding is often an active, shape-shifting process that facilitates catalysis. So to summarize, enzymes lower activation energy by binding their substrate at the active site, and they form a highly specific enzyme-substrate complex. This interaction can occur through a rigid, pre-shaped mechanism like the lock and key model, or through a more flexible dynamic mechanism like the induced fit model. Next, we're going to talk about another important element of enzyme function, and that is the fact that some enzymes can't operate alone. They need helper molecules to assist in catalysis, and these include cofactors and coenzymes. Both cofactors and coenzymes are non-protein molecules that help enzymes do their job, but there is a key difference between them that we need to be able to recognize. Cofactors are generally inorganic molecules, often metal ions like magnesium, zinc, or iron, and they are often obtained through the diet as dietary minerals. And they're usually going to participate directly in the enzyme's chemical reaction. For example, they can help stabilize negative charges, or they can assist with electron transfer. Here, there's some definitions we need to know. There are apoenzymes and there is holoenzymes. Apoenzymes are enzymes without their cofactors. Holoenzymes are enzymes with their cofactors. Then there's coenzymes. Coenzymes, on the other hand, are small organic molecules. Most coenzymes are derived from vitamins, and some of the most common examples include NAD+, FAD, and coenzyme A. These molecules often carry functional groups or electrons from one reaction to another. So they act kind of like temporary carriers in metabolic pathways. So when you see an enzyme that's inactive on its own, but becomes functional in the presence of a vitamin or metal ion, chances are you're looking at an enzyme that requires a cofactor or a coenzyme. Now that we understand how enzymes function, how they interact with substrates, and what additional molecules they may need to be effective, we can now move into the next major objective on enzyme kinetics. Enzyme kinetics is the study of how fast enzyme-catalyzed reactions occur and what factors influence that rate. Two major factors are the concentration of the substrate and the concentration of the enzyme. Now, other environmental conditions like pH and temperature also matter, but in this objective, we're going to focus on how substrate concentration affects the rate of reaction. If we imagine gradually increasing the amount of substrate available to an enzyme, we're going to see that the reaction rate will go up. Why? Well, because more substrate molecules means more opportunities for the enzyme to bind and convert them into product. However, this increase in rate doesn't continue forever. At some point, every active site on every enzyme is occupied, and this is known as saturation. Once saturation is reached, The enzyme is working as fast as it possibly can. This maximum speed is called Vmax. This is the maximum velocity or reaction rate that the enzyme can achieve under these conditions. Adding even more substrate after this point won't increase the reaction rate because all of the enzymes are already busy. To describe this relationship mathematically, we're going to be using the Michaelis-Menten equation. It helps us understand how reaction rate changes as substrate concentration changes. And the equation that we're going to be using here is shown in this part. Here, V is the rate of the reaction. S in brackets, this is the concentration of the substrate. Vmax is the maximum possible rate when the enzyme is saturated. And Km is the Michaelis constant. We're going to define this more precisely in just a moment. But this equation tells us that at low substrate concentrations, the rate increases sharply with small increases in substrate. But at high substrate concentrations, the rate levels off and it's going to approach Vmax. Now, one of the most important concepts from this equation is Km. Km is defined as the substrate concentration at which the reaction rate is equal to half of Vmax. So if you graph the reaction rate versus substrate concentration, Km is the X value where the enzyme is operating at 50% of its maximum speed. We can derive this by plugging V equals Vmax divided by 2 into our Michaelis-Menten equation and then solving for S. When you do that, you're going to find that Km equals S under those conditions. Now, what does Km tell us about the enzyme? It reflects the affinity of the enzyme for its substrate. So a low Km means the enzyme reaches half max velocity at a low substrate concentration, and that implies high affinity. A high Km means it takes a lot of substrate to get the enzyme working at half speed, which means lower affinity. Another important parameter is Kcat. This is known as the turnover number. This tells us how many substrate molecules one enzyme molecule can convert into product per second when the enzyme is fully saturated. We can go ahead and relate Vmax to Kcat using the equation you see right here. All right, in this equation, E is the concentration of the enzyme. So this tells us that the maximum rate depends both on how much enzyme is present and how fast each one works when it's saturated. If we plug this expression for Vmax into the Michaelis-Menten equation, we get the equation that you see here. At a very low substrate concentration, specifically when Km is much greater than S, this equation can actually be simplified. Since S is so small compared to Km, we can actually drop the concentration of the substrate, S, from the denominator, and we end up with this equation right here. This simplified form is often used to describe reaction rates when substrate is limiting. So here, Kcat divided by Km is referred to as the catalytic efficiency of the enzyme. So a large Kcat or a small Km is going to result in a higher catalytic efficiency, which indicates a more efficient enzyme. So to summarize, the Michaelis-Menten model helps us describe how substrate concentration affects reaction rate. Vmax tells us the upper limit, Km tells us about substrate affinity, And kcat tells us how efficient each enzyme is when it's fully active. Now, there is another tool that can be used to analyze enzyme data when determining values for Km and Vmax, and that's the Lineweaver-Burk plot. The Lineweaver-Burk plot is a double reciprocal graph of the Michaelis-Menten equation. What that means is instead of plotting reaction rate versus substrate concentration, we take the inverse of both. So specifically, we plot 1 over velocity on the y-axis and 1 over substrate concentration on the x-axis. This turns the hyperbolic Michaelis-Menten curve into a straight line, which makes it easier to extract key values. Here, the y-intercept of the Lineweaver-Burk plot gives us 1 over Vmax. The x-intercept gives you -1 over Km, and the slope of the line equals Km divided by Vmax. The final concept we want to discuss in this objective is cooperativity. Some enzymes don't behave in a simple way when it comes to substrate binding. Instead, they show cooperative binding, where the binding of one substrate actually changes the enzyme's shape in a way that increases the likelihood that additional substrates will bind. So in other words, binding in one substrate makes it easier for the next one to bind. And this behavior is typically associated with enzymes that have multiple subunits or multiple active sites. And it reflects a transition between a low affinity state, sometimes called the T state, and a high affinity state, sometimes called the R or relaxed state. We can quantify this cooperative behavior using something called Hill's coefficient. Now, all you need to know here is what the Hill coefficient means when it's compared to 1. If the Hill coefficient is greater than 1, then the enzyme shows positive cooperativity. So binding one substrate makes others bind more easily. If the Hill coefficient is less than 1, that's negative cooperativity, meaning binding one substrate actually makes further binding harder. And if the Hill coefficient is equal to 1, then there's no cooperativity, and the enzyme follows traditional Michaelis-Menten kinetics. With that, we can now move into the next objective. Here we're going to cover the effects of local conditions on enzyme activity. Enzymes work under very specific conditions, and small changes in temperature, pH, or salt concentration can dramatically impact their ability to function. So let's walk through each of these environmental factors. First, let's talk about temperature. In general, enzyme catalyzed reactions tend to double in speed for every 10 degrees Celsius increase in temperature. This is because molecules are moving faster, they're colliding more frequently, and they're overcoming activation energy more easily. However, this increase in activity only continues up to a certain point called the optimal temperature. Beyond that, the enzyme begins to denature. And denaturation means that the enzyme's three-dimensional structure starts to unravel, especially the shape of the active site. And once the structure is lost, then the enzyme can't function. So while a little heat helps, too much can shut everything down. Next is pH. This also plays a major role in enzyme activity. Each enzyme has an optimal pH range, and this varies depending on where the enzyme normally operates in the body. So for example, pepsin, which is a digestive enzyme in the stomach, it works best at very low pH. While trypsin, this is found in the small intestine, it prefers a more basic environment. So pH, it can affect enzyme activity in two major ways. One, it can alter the ionization state of amino acids in the active site, and that can disrupt substrate binding. Or more drastically, extreme pH values can cause denaturation, just like high temperature can, and this disrupts ionic bonds and hydrogen bonds within the protein. Finally, let's discuss salt concentrations. These are really important. Increasing salt levels can disrupt ionic interactions and hydrogen bonding as well within the enzyme or between the enzyme and substrate. And these disruptions can really alter enzyme shape, and that can decrease activity. Now, while this isn't a large factor in vivo, it's something that scientists have to carefully control in the lab when they are attempting to study the effects of salt on enzymes. So in summary, enzymes tend to work faster as temperature rises until they overheat and denature. pH has to be just right, not too acidic or too basic, because that can destabilize the enzyme. And then high salt concentrations in vitro can disrupt key bonds and change how the enzyme functions. Now that we've seen how environmental conditions can affect enzymes, let's turn our attention to regulation, which is our final objective for this chapter. Enzymes, they do not operate in isolation. They are tightly regulated within metabolic paths to make sure that reactions occur when and where they're needed. And one major form of control is called feedback regulation. In feedback regulation, products from later steps in a pathway regulate enzymes that act earlier in the sequence. For example, when a product builds up, it can signal the pathway to slow down or stop. And this is called negative feedback or feedback inhibition. It helps prevent the cell, for example, from wasting energy making too much of something it already has. There's also something called feed-forward regulation, where an intermediate earlier in the pathway activates an enzyme that comes later, that's essentially preparing the system in advance when more product will soon be needed. Now, in addition to regulation through pathway feedback, enzymes can be regulated by molecules that inhibit their activity. These inhibitors may bind reversibly or irreversibly, and here we're going to focus on 4 types of reversible inhibition. The first is competitive inhibition. In competitive inhibition, an inhibitor binds directly to the active site of the enzyme, and it physically blocks the substrate from entering. Since both the inhibitor and the substrate compete for the same binding site, this type of inhibition can be overcome by simply adding more substrate. So that means the enzyme can still achieve the same maximum reaction rate, or Vmax, but it now requires a higher concentration of substrate to reach that speed. So Vmax stays the same, but Km increases. And the increased Km reflects the fact that the apparent affinity for the substrate is lower due to competition. So on a Lineweaver-Burke plot, lines are going to intersect on the y-axis for competitive inhibition because Vmax stays the same while Km increases. Next is non-competitive inhibition. In this case, the inhibitor binds to a different site on the enzyme, an allosteric site, and not the active site. And this binding changes the shape or function of the enzyme so that it can't perform catalysis effectively, even if the substrate is still able to bind. Because the inhibitor isn't interfering with substrate binding, the Km stays the same, but the maximum reaction rate, Vmax, it's going to decrease because there are fewer functioning enzymes that are available to perform the reaction. So on the Lineweaver-Burke plot, the lines are going to intersect at the x-axis because Km stays the same, but Vmax decreases. Then there's uncompetitive inhibition. This is a bit different. In uncompetitive inhibition, the inhibitor binds only to the enzyme substrate complex. It does not bind to the free enzyme. And this locks the substrate in place and then prevents the reaction from proceeding to product. As a result, both Km and Vmax decrease. The Km decreases because the enzyme appears to have a higher affinity for the substrate. Vmax decreases because the inhibitor prevents the enzyme from completing the reaction. So on the Line-Weaver-Burke plot, the lines appear parallel because both Km and Vmax decrease proportionally. The last type is mixed inhibition. This can either bind to the free enzyme or to the enzyme substrate complex. Let me repeat, a mixed inhibitor can bind to either the free enzyme or to the enzyme substrate complex, but with different affinities. Now, like non-competitive inhibitors, mixed inhibitors are going to bind to allosteric sites, and they are going to affect both the Vmax and the Km. Vmax is going to always decrease since inhibition reduces the enzyme's functional capacity. But the effect on Km depends on where the inhibitor prefers to bind. If it prefers the free enzyme, then Km increases. If it prefers the enzyme substrate complex, then Km decreases. So the overall effect on Km depends on the relative binding affinity. If we summarize these 4 types of reversible inhibition in a comparison table, here's where they would differ. For competitive inhibition, the inhibitor binds at the active site, and what you'll see is that Km increases and Vmax stays the same. For non-competitive inhibition, the inhibitor binds at an allosteric site. Km stays the same, but Vmax decreases. For mixed inhibition, the inhibitor also binds at an allosteric site. Vmax decreases, and Km may either increase or decrease depending on binding preference. Finally, for uncompetitive inhibition, the inhibitor binds only to the enzyme substrate complex at an allosteric site. Both Km and Vmax decrease. Now, in contrast to these reversible mechanisms, irreversible inhibition involves permanent deactivation of the enzyme. This usually occurs when an inhibitor forms a covalent bond at the active site or otherwise causes structural damage that the enzyme cannot recover from. The active site is going to be either blocked or destroyed, and the enzyme cannot function again. The only way for the cell to regain that activity is to synthesize new enzyme molecules. We also need to mention a few other regulatory mechanisms that don't involve inhibitors at all. Some enzymes are regulated allosterically, meaning that other molecules can bind at sites other than the active site to activate or inhibit enzyme activity. And these activators and inhibitors shift the enzyme's shape and alter how effectively it works. Another mechanism is covalent modification, where the addition of chemical groups changes the enzyme's shape, activity, or stability. Finally, some enzymes are produced in an inactive form called zymogen. These are activated only when needed, usually by cleavage of part of the protein. And this is a way of storing potentially dangerous enzymes in a safe form until the correct time or location arises. And with that, we're at the end of our enzyme chapter. We've covered everything we need to know. I hope that this has been helpful. Please let me know if you have any questions, comments, concerns. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.